Bhagyalakshmi ma'am. Okay. So see in the board exam, board Telugu Academy textbook, total 8 units are there. Like first one, diversity of living world. Second unit is structural organization. Third unit and fourth unit, animal kingdom, both invertebrate and vertebrates. Fifth unit, protozoa, reproduction and locomotion is there. Sixth unit, human health is there. Seventh unit, cockroach is there. Eighth unit, ecology is there. Again under ecology, 4 lessons: organisms and populations, ecosystem, biodiversity, environmental issues. Total 8 topics. Okay. Among them, first one is deleted because same points are covered also in botany. Fifth one is deleted because at several places we study the type studies of protozoa. Even cockroach is deleted. Ecosystem is deleted, environmental issues deleted. Environmental issues means pollution. Already pollution lesson is covered in chemistry also. So the topic is removed from zoology. It is in chemistry. Ecosystem is also deleted. So, so far we have completed the total animal kingdom, and we have completed all the tissues, coelom, symmetry, and all. Human health perfectly we have completed. That all the immune system, cancer, types of immune systems, all the parasites, life cycles, adaptations in the parasites, adaptations in the host, detailed everything we have completed. Even organisms and populations detail we have completed. Now only one lesson left over, that is biodiversity. So today we are going to I'm going to start new lesson, biodiversity and conservation. Biodiversity and conservation, all of you. So I think you understood. These are the units in the Telugu Academy. So what are deleted according to, it is already you only posted to me, and online it is available. What are deleted? So these red ones deleted, and the remaining topics we have completed in detail, everything perfectly we have completed.
Yeah, ma'am.
Animal kingdom, tissues lesson we have completed. Yeah, yes. Once the biodiversity is completed, your syllabus will be completed. Then they'll start junior inter, this one, according to NCRT, junior inter topics like digestive system, respiratory system, circulatory system, excretory system, musculoskeletal system. Yeah, yeah, NCRT also we have to go through. You're not going to give any board, CBSE board exam, but continuity will be there so that they'll complete your topics as early as possible. 2, 3 revisions will be covered, revised on the base of, yeah, removed. Nah, it is there in the botany also. So here it is removed. Okay, so as early as possible, human physiology will be started, and in the March exams are there. So maybe they'll stop by January like that. So February for preparation. But it, what do you mean by NCRT also? What do you mean? Wait, I'll open your mics. It's not possible to complete NCRT now itself, but they start continuation. Maybe in February month, put it on hold and allow you people to prepare for board exam. Again, we'll start continuation of the second year. So generally, what is the practice? In the second year, by September, we complete the portion. October, November, December, January, 4 months. It's a revision program. Again, both the first year, second year topics are revised. 2 times revision will be there. 2 times. Most probably this time September onwards. 5 months will be there. Again, they repeat. All the topics revision will be there. It helps you for your NEET exam preparation. Okay. So let us start this lesson, new lesson, biodiversity and conservation. Put heading all of you. So we're going to study meaning and definition. Next, levels. Under levels, genetic diversity, species diversity, ecosystem diversity, also called as ecological diversity. Among ecosystem diversity, again, three types are there: alpha, beta, gamma. So we are going to study them, all of you. Page number 114, 114. Page number 114. Page number 114. Written. Completed. Now start the meaning. What do you mean by diversity? Simple. Variety of life on earth. What's the meaning? Variety of life on earth. Variety of life on earth. Okay. So what's the definition? We are going to study. We are going to study variation of life and biodiversity. What's the definition? Variation of life at all levels. At all levels. At all levels. Within a species. Within a species. Within species. Among species. Among species. And among ecosystems. Among ecosystems. So, what we are going to study now? Variation in the life at all levels. All levels means within a species. That is genetic diversity. Species diversity, that is among species. And also among ecosystems, you are going to study the diversity. Variations of life, we are going to study. So, what is the definition? Perfect definition. Enter long kakunda. Another definition. Totality. Totality. Please keep off your mics when you are not using your mic. Keep off your mic. Totality of genes. Short definition it is. Totality of genes. Okay. Species and ecosystems. Totality of genes, species and ecosystem of a region. Biodiversity means what? totality of genes, species, and ecosystems of a region. It is second definition. The main definition is variation of life, changes, different varieties of life at all levels, at species level, among the species, among the ecosystems. So we are going to study different varieties, how varieties are formed, what is the use, okay, like that in this lesson. So one important thing, one other important point, this term diversity was popularized by important term bio, sorry, biodiversity, popularized by, I said popularized by, popularized by Edward Wilson, important, Edward Wilson. Edward Wilson, all of you. Completed? Edward Wilson. After going through the two definitions and the name coined by Edward Wilson, what is the meaning? That is nothing but variety of life on earth. Now start with the levels. Did you complete this meiosis and mitosis in the botany?
Yes,
Yes,
ma'am.
ma'am.
Okay, good. That's enough. I need that, whether you have completed or not. Now see the first one. See the genetic diversity. Now start the levels. Levels of biodiversity. Start with the first one, genetic diversity. There is variation between individuals, individuals of a species, of a single species, that is intraspecific within the species. Intraspecific. May I tell one example, you'll understand. Take our human. We are all same species. Homo is the genus name.
Sapien.
Yes, is the species name you know very well. And we are all same species. Sapiens means intelligent animal. But among ourselves within the world, Africans, Negroid. But of course, for human, we don't use the term variety. We should not use the term variety. But for human, we should use the term race. You know very well, Negroid race. All the Africa, countries of continent South Africa, like Sudan, Libya, South Africa, Egypt. You know very well how they look. Dark skinned, heavy body, muscular body, short and curly hair. By looking at them, you can understand, okay, he is a Negroid. Now move to the Tibet, Nepal, Mongolia, Japan, Korea, Indonesia, Thailand. All here you could see by looking at them, you say Mongoloid race. Mongoloid race. Now, when you come to America, Europe, Australia, Caucasoid race, like that, we human, within our human, several races are there. The best well known races I told you just now, three varieties: Caucasoid, Mongoloid, Negroid. Means within the species, three varieties are there, three races are there. So there is intraspecific variation between the individuals of single species. Try to understand. If you understand human, it is easy to understand here. I think all of you, is it clear now?
Yes, ma'am.
So it is
Yes, ma'am.
intraspecific. Yes, now continue. Yes, now important. It is developed due to how it is developed. First thing important, that is produced due to, or you can say developed due to. And these gene variations developed is important due to mutations, gene mutations. Mutation means change in the gene. Gene mutations, changes in the gene. Gene recombinations. Gene recombinations. Now tell me, completed DNA structure,
Yes, ma'am.
chromosome structure,
Yes, ma'am.
Yes, ma'am.
RNA ribosomes.
Yes.
Yes,
Yes,
ma'am.
ma'am.
Now let me say, explain you. Listen to me, all of you. All of you. First of all, try to understand what is gene. A stretch of DNA. We have, you have 2 meters long DNA in every cell in the nucleus. 2 meters.
1 meter
One
maternal, 1 meter
meter, yeah,
paternal.
1 meter from mother and 1 meter from father. 1.1. 1.1 meter from mother and 1.1 meter from father. That all 2 meters long DNA is not a gene. 30,000 genes are there in human body, in every cell. But all 2 meters is not a gene, but a stretch of DNA. Listen to me. A stretch means a length of DNA. Say suppose this is one particular length of DNA, not 2 meters, a small stretch, a particular stretch of DNA, which carry information for mRNA, means which can transcribe. I think you know the formation of RNA from DNA is called transcription.
Transcription.
Yes. One can transcribe into mRNA, and this mRNA, along with the ribosomal RNA, ribosomes
Polyzoom.
Holism.
RNA, and according to the code on the DNA, tRNA brings particular amino acids, and amino acids together form protein. So all the three RNAs, what are the three RNAs? Let me show you. mRNA, mRNA participating protein synthesis. Proteins. It produces proteins. Evaru, mRNA. How? Ribosomal RNA stands on the code rRNA, means ribosomes. I think you might have studied their workbenches for protein synthesis. Like, see now,
Yes, ma'am.
yeah,
Factories for that.
my laptop, all the books, everything I arranged on the table. Without this table, it's not possible to me to focus completely, standardly to focus the camera onto the board and to put my books, what I am going to explain, everything I prepared. It's a workbench here for me. A table is there. In the same way, ribosomes provide a table where protein synthesis can be done, even in the kitchen. To process all the food, we put a platform in the kitchen, isn't it? dining table, working table, office table, like that. To process our work, we always require a bench. So they provide a bench so that protein synthesis can occur. And according to the code language on the mRNA, tRNA brings amino acids, and all amino acids are read continuously. Amino acid A stands for amino acid here. So polypeptide chain of amino acid, nothing but protein. So all three RNAs participate: mRNA, rRNA, and
Yeah.
what I have drawn is tRNA. Clover leaf model. I think you might have studied. Is it completed or not? Clover leaf model. Have you studied?
No, ma'am.
No,
No,
ma'am.
ma'am.
No, ma'am.
Okay, just I have drawn like this. It stands for tRNA. You'll study it afterwards. Detail. tRNA. So anticodons will be there, and they read the code here. And according to the code here, amino acid is brought. And all amino acids are arranged together. Polypeptide chain of amino acids is nothing but protein. So, what all the three RNAs helping in to produce a protein? So protein may be a hormone, like example growth hormone. Protein may be an enzyme, like your pepsin enzyme, trypsin enzyme, to digest the food material in your body, what you're eating. Maybe they are hormones already. I told you structural proteins like keratin protein, collagen protein, elastin protein. Already we are done constructing your body. Are you following me? Constructing your body, performing the regulating the functions of your body, helping in all the metabolic activities, regulating the activities, constructing the structure. So these proteins are maintaining your life and constructing your life. And proteins are formed on RNA. RNA is formed on DNA. And DNA stretch, which is carrying this information, is nothing but a gene. Got it now, all of you? So
Yes,
suppose
ma'am.
imagine my earphones wire. Say this is 2 meters long DNA. Got it? Enclosed inside the nucleus. All these 2 meters long DNA is not carrying gene. Of course, stretch it out. Say this is 2 meters long DNA, stretch it. So all this 2 meters long is not carrying gene. What do you mean by gene? Which carry information is a gene. Suppose from here to here, this small stretch is carrying information to produce mRNA, then it is said to be gene. So a particular stretch of DNA which carrying information for an mRNA, okay, which can produce protein is gene. Is nothing but it is gene. All of you.
Yes, ma'am.
Okay. So what happened? What's the problem here then? If any change in the code language, any change in the code language,
Mutation occur.
ఒక్క నిమిషం ఉండండి నాన్న. Change in the code. Automatically it changes code in the mRNA. If mRNA code is changed, code means nothing but DNA is made up of adenine, guanine, cytosine, and thymine. So their arrangement is changed. Automatically changes the code in the mRNA. If mRNA code is changed, amino acid is changed. And now, if new amino acid is added, automatically protein loses its shape and structure. Once it loses its shape and structure, functional capability is also changed. A new protein is formed. Maybe a new enzyme is formed, or the old enzyme lost its function, like that. So changes occur. which bring diversity, variations, different varieties. So changes in the DNA is called mutation. Like best, let me explain you one idea I'll give you. One idea I'll give you. Suppose an mRNA, if it is guanine, adenine, guanine, GAG. If it is code on mRNA, guanine, adenine, guanine, then automatically tRNA brings glutamic acid. Glutamic acid is an amino acid. How mRNA cancels GAG? Automatically DNA should be CTC. So in the DNA, code is CTC. Then on the mRNA, GAG. Then automatically tRNA brings glutamic acid. Now let me explain the mutation now in the DNA. Okay. Now let me explain what is mutation. Suppose, of course, here I am giving a bad mutation. Mutation may be a good one. Okay. Let me show you what. Suppose change in this thymine in the DNA. I'm bringing change here. I'm explaining change here, all of you. Yes. Now it is CAC. Means instead of thymine, it is changed. Point mutation. Nitrogen base changed. Adenine. Then automatically in the mRNA, it is GUG. I think you know instead of thymine, mRNA
Uracil.
yes,
Uracil.
then it is GUG. Uracil is there. Instead of adenine, it is uracil. Instead of adenine, it's a point mutation, it is uracil. Then automatically tRNA can't bring glutamic acid, but it is valine amino acid. Instead of glutamic acid, valine amino acid is brought. So simple change, small change, very small change. So we named it as point mutation. Then what's the problem? Glutamic acid is replaced by valine. Then what's the problem? Instead of normal RBC, these people who are having this change in their beta globulin, instead of normal donut shaped RBC, happily the donut shaped RBC. ఇలా ఉంటుంది కదా ఆర్బీసీ బై కాన్కేవ్ బోత్ సైడ్స్ ఇట్లా ఉంది ఆర్బీసీ of course it is blue color, just imagine red color RBC like this. Instead of like this, RBC membrane is broken and it appears like this. All the hemoglobin inside the RBC are released and RBC attain a sickle shape. Sickle means the tool with which we cut grass. Sickle shape. Why? Simple, small change, one amino acid changed. Why? Small change. Instead of adenine, it is uracil. What is the problem? Small change in the DNA. Instead of thymine, it is adenine. Very small change, Nana. But it leads to a genetic disorder, sickle cell anemia. If from both mother and father side they got that mutation, they can't live more than 14 years, they will die because of that disorder. So try to understand, sir. I said already in the beginning, I'm going to explain a bad mutation, which leads to disorder. Understand, mutation means a small change in the DNA, can bring change in the code of the mRNA, can bring change in the amino acid. One amino acid is also changed. Shape and structure is changed. One shape and structure is changed. Lock and key lost. So functional ability is lost. So it can bring a new disorder, maybe a new character, or maybe a genetic disorder. Sometimes that disorder leads to death also. So gene mutation. If it is a good mutation, variety will develop, diversity. You know, in within our India, 1,000 varieties of mangoes are there. Every summer in our Hyderabad, they conduct exhibition of mangoes. 500 mangoes displayed over here. So how these different varieties developed? Change in the DNA. I think you understood gene mutation, all of you.
Yes, ma'am.
Yes, ma'am.
Now next step. Gene recombination. Yes, gene recombination means what? Let me show you. And it's a natural process, very regular, normal process. You already studied meiosis. Meiosis occurred during gamete formation. Kada. Some people pronounce it as meiosis. Yeah, and it occurs during gamete formation, means during sperm formation from the paternal parent, father, and egg formation from the mother. So how it occurs? Chromosomes stand. Kada. During gamete formation, chromosome from mother side. Of course, female we always represent with pink color. I'm giving red color here. Blue for boys. I'm using blue color. You know very well, tetrad formation occur during prophase 1, meiosis 1. I think you have studied tetrad
Yes, ma'am.
formation. Sister chromatids are formed. And the inner non-sister chromatids crossover. Have you done?
Yes, ma'am.
Yes.
Combination of the
Crossing over, yes. And the point where
combination.
they cross it is called
Recombination of the
plasma. And what happened? Genes shuffle. Kada? Genes from the mother's side, they shift on to the father's chromosome. Yes, here. And genes from the father's side can shift on to the mother's chromosome. And they separate. Yes, suppose this chromosome in the egg, mother's egg, అనుకుందాం ఇది egg అనుకుందాం. So, dadima, కాదు dadima కాదు. Maternal grandmother is called nanima kada, nanima kada. Nanima chromosome with the Nana G genes, few Nana G genes. Got it. And another egg, Nana G chromosome perfectly dominated with the genes from Nana G chromosomes with Nanima combination. And from which egg we are formed, it depends. Out of four, only one egg is undergoing fertilization. Suppose this egg is fertilized. In the same way, maybe from father's side, Dadima genes with few Dadaji's combinations. So you are born with four combinations. Okay? Amama, Amama Tata. Amama means Nanima, Nanima husband, Nanaji. Dadima, Dadaji. Four grandparents combination you are. So recombinations occur during gamete formation and it's a natural process. So every time you are a combination of four parents, I'm sorry, four grandparents, four parts of your grandparents. But nowadays we are not much more interested in our grandparents, putting them in the old age homes. But you are the combination of four grandparents, remember that. Okay, back to the lesson. So, gene level mutation and at chromosome level recombinations, even new varieties are formed. New varieties are formed due to environmental changes. Yes, variations, variability. Means if you are in the tropical region, generally you are dark. If you are in the polar region, you are fair, like that. So, environmental variability, all these things develop changes at the gene level, bring that changes at the gene level, all of you. Yeah, and very important, last important point, because you'll get statement type question here. Yes, variation between individuals of single species means within a species intraspecific. That changes develop at the gene level due to mutation, at chromosomal level due to recombination during gamete formation, due by meiosis, gene recombinations, and even at environmental level brings the variability as we adapt to to live in that particular place. So we bring some changes in our body. Okay. Now, important last point. Important it is. I'm giving star rating. Yes, genetic diversity. You'll get statement type question. Genetic diversity. Genetic diversity is advantageous for survival. Just now someone typed Anjum, I think. She's typed adaptation. Yes, it helps us to adapt to that environment and to survive in that place immediately. And it is the basis of next important point. It is the basis of formation of new species. Formation of new species means what we call that process as speciation. Formation of new species called as speciation. అయిపోయింది. Very, very, very important points. I put it on the board. All of you note down. Fast, Nana. Completed? Completed fast.
Yes.
Yes, ma'am.
Ma'am.
So go through the example given in your textbook, given in your material, like Raulfia matoria is a plant. Excuse me, I'm sorry. Plant name of Himalayan range. Several plant varieties are there. Same species, of course, but several varieties are there. Based on the chemical we extract from it, that chemical is reserpine. Reserpine is an alkaloid
It's an angle.
we extract from that plant. And we use this alkaloid, that chemical extracted from the plant, to treat blood pressure, particularly high blood pressure. What is the plant name? Raulfia omatoria. Okay, in the Himalayan range we get that, and what the scientist observed, several variety are there. Depending upon the amount we extract, some plants can give 5 grams, some plants can give 10 grams, like that. Amount how much we can extract. At the same time, potency of the chemical. Potency means capacity, how strong it is helpful to treat that blood pressure. Potency of the, I think you know the potency of the medicine, capacity of the medicine, dose, then working capability. So depending upon the amount we extract, potency of the drug, different varieties are there. How they develop? At gene level mutations. At the same time, example, 50,000 varieties of rice varieties are available in India. 50,000. మనకి రెండు బాగా తెలుసు, హైదరాబాద్ పీపుల్, బిర్యానీ రైస్, that aroma, that specific taste and smell is only we could see in that biryani rice. And normal rice, everything every day mama cooks, that is a different rice, or namasuri. Only two I could say in the class. 50,000 are there in India. As already I told, 5,000 variety of mangoes are there. Roses, so many varieties of roses, you know. 1,000 varieties of mangoes, and just imagine like Banginpalli and Totapuri, Rasam, from which we get juicy mangoes. Okay, red color base Totapuri mangoes, like that. Okay, Nana. So 1,000 varieties of mangoes. All these are same species, but varieties developed due to gene level change. Now come to the species diversity. Next. Yes, species diversity means different varieties of species, different varieties of species living in a given area. Different varieties of species living in a given area. Now you have to take a particular area, given area. Got it? Number, variety you have to count like parrots, pigeons, sparrow, crow, peacock, like that. Now here itself, I counted five varieties of five different species of birds: dogs, cats, wolves, cheetah, uh, tiger, lion, leopard. Seven varieties of carnivores I counted here itself. Like that, different varieties of species living in an area. I think important, Nana, how we will understand the species are rich in that area. You have to consider two things to say species. That area is good in species diversity. What are the two things important point it is? First thing is that species richness. How could you say that area is good in species diversity? What you have to check? Species richness. Richness means number of variety. At the same time, you have to count evenness also. Evenness. Evenness. N ఇంకొక N రావాలి. Evenness. Okay. I'll show you one picture to say both the richness as well as evenness. You have to count both of them. You can't say only lion is there, tiger is there, wolves are there, cats are there, dogs are there, cheetah is there, leopard is there, all these are carnivores. You can't say that. It's not enough to say it is good in biodiversity, rich in diversity. But at the same time, you have to count the evenness also. Evenness means the number of distribution should also be uniform. I'll show that picture, then I'll stop tomorrow. We'll continue with the ecosystem and, of course, continuity of this topic. Let me show you. I think it takes time. Let me complete. Like within your book, the topic is also given. Let it open. ఇక్కడ ఇచ్చినాడు చూడండి. 3 diagrams are given. In the first diagram, pigeon, sparrow, and parrot are there. Three varieties are there. Wait a minute, I'm coming back. One minute, I'm coming. What happened to the diagram where it has gone? ఒక్క నిమిషం నాన్న. Yes. Now check this diagram, all of you. Because you'll get question on this topic. Is it there on the screen? Copied or not? Okay, I'll do one thing. Tomorrow I'll put it on the screen and I'll show you tomorrow.
Okay, ma'am.
Okay, ma'am.
Thank you, ma'am.
Okay, you can leave now. Tomorrow I'll
Thank
continue.
you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Have a good day.